morning everyone. Welcome to the second session of last year of our workshop. In this session along with us, Dr. Urvish Chaya. Dr. Urvish Chaya did his master and PhD in the subject of microbiology from Sardar Patel University. Currently he is working as assistant professor in a department of microbiology at NV Patel College, Vallabh Vidyanagar. He possesses a teaching experience of 11 years and he has published more than 10 research papers, more than 15 research papers in a journal of national as well as international review. He has completed 2 research projects from National Funding Agency and he possesses a specialization in a subject of microbiology with special emphasis on bacterial genetics. Sir, I request you and welcome you on behalf of Ramakrishna Institute as well as GSPK.
Let me first convey my courtesy to Dr. Darshan Marjadi because my association with Darshan sir is not recent one. We are associated with each other since very long and we share some common thoughts, we share some common ideology, and that's why I am here today on his personal request to me. So without wasting much time, we will come to our topic. As far as the competitive examinations are concerned, there are certain topics which you cannot avoid. You may like or you may dislike them, but you may not avoid them. For example, bacterial genetics or the microbial genetics. The reason is that if you look at the eligibility criteria for JNU CBE, students from microbiology, biochemistry, biotechnology, zoology, botany, even chemistry and MBBS, they all are eligible for this examination. So there are certain topics which are overlapping in all these courses. For example, bacterial genetics, or you can say microbial genetics, enzymology, biomolecules, etc. So these are the topics which you cannot avoid when you want to prepare for the examination for JNU CBE, or even if you want to prepare for NET, CSIR NET, or any other examination. So that's why, as far as the competitive examinations are concerned, earlier speakers must have told you that there are 2 different kinds of questions which can be asked by and large. One is those type of questions which are information based, where you need not to apply any rational or you not need to apply any logic. And second category of questions are those where you need to require application of logic or the rational. So in today's presentation, I have divided this presentation into several topics, and this is followed by certain model questions which have been taken from the previous year's JNU papers, and we will try to solve them. So that's why when it comes to the application of the logic, it is obvious that if you are not aware of the principle of that particular topic, you may come up with the wrong answer. And it is possible that that wrong answer may be the part of the 4 different options which has been given, because these MCQs are very meticulously designed. For example, let me give you one. For example, महाभारत જે છે, महाभारत ની અંદર આપણને માતા કુંતીનું પાત્ર ખબર છે, हम जानते हैं. એવો ક્વેશ્ચન દાખલા તરીકે પૂછાય કે માતા કુંતીને પુત્રો કેટલા હતા? 4 options are given: 3, 4, 5 and 6. So when you read the question, first of all you will recollect and you will say પાંડવો તો 5 જ હોય ને, એટલે you will tick mark 5. પણ પછી આપણને રિયલાઇઝ થાય કે ના, કર્ણ પણ કુંતીના જ પુત્ર હતા, એટલે તમે 6 ટિક માર્ક કરો. You will come to the answer 6. But then you realize that na, Sadeva and Nakul to Madrina putra hota. So the correct answer is 4 actually. So that's why These kind of logic and rationale needs to be applied. Now, let us go to the some of the basic concepts. Since you are from the 6th semester, yeah, right? You are from the 6th semester, all of you. So you must be knowing all these things, some of the basic concepts. But we will just go through this. We know that the genetics, it is the science of hereditary study of what genes are, how they carry the information, how they are replicated, passed along, and how expression of the information determines the characteristic of the organism. And the genome means all the genetic information into a cell or the chromosome, which is organized in a unit of the genome, that is the bundle of the DNA bacteria. Generally bacteria has one chromosome, humans have 46, we know, and yeast has 7 chromosomes. Genes, they are the segment of the DNA that code for the functional products like rRNA, tRNA or the proteins. So when we talk about the genes and when we talk about the mutation within the genes, we always focus on the DNA. When we define the mutation, we say that the change in the nucleotide sequence is known as the mutation. Change in the nucleotide sequence is known as the mutation. But if the change in the nucleotide takes place in the RNA, then it is not going to be inherited into the subsequent generations, because the DNA is the main replicating or the genetic material which is inheritable. So that's why when we talk about any event, genetic event related, then we must understand that this must be related to the DNA. For example, mutation. When we say mutation, change in the nucleotide sequence, it means the change in the nucleotide sequence in DNA. If it is in the RNA, then it will not be inherited, it will not be percolated into the subsequent generation. So that's why it is not a mutation actually. Are you getting it? Okay. So as far as the genomics is concerned, it is the field of the genetics involved in the sequencing and molecular characterization of the genome. Some examples are given. That E. coli, it has the 4 million base pairs, which has 3 to 4,000 genes. Yeast has 12 million base pairs, corresponds to 5 to 6,000 genes, and humans, they have 3 billion base pairs corresponding to 30,000 genes. Now, based on this information, some questions can be asked in the competitive examination. For example, if the organism has 4,200 base pairs, how many helical turns that DNA molecule will possess? Any idea? For example, if the given DNA molecule, this kind of information, why it is important? Because from these questions can be asked. For example, if you have the DNA of 4,200 base pairs, this is the DNA present within the organism. How many helical turns that DNA will be having? Very simple question. 10. Very simple question. This is very simple question. You should know what is the physicochemical property of the DNA. You know that one helical turn contains 10 base pair of DNA. So you simply divide this 4,200 base pairs with the 10. Yes or no? If 10 base pairs produce one helical turn of DNA, 4,200 base pairs produce how many helical turns? Very simple. 420 helical turns will be there in the DNA structure. Are you getting it? Similarly, if I want to find out what will be the length of this DNA, what will be the length of this DNA in terms of the nanometer or micrometer or meters, how can I find out? 4,200 base pair, 420 helical turns. Now what is the pitch size of one helix? What is the pitch size of one helix? I think you are aware with all these facts. Yes or no? What is the pitch size of that one helical turn of the DNA? 0.34 nanometer. 0.34 nanometer. It means the one helical turn of the DNA, which is having 10 base pairs, is having the pitch size of 0.34 nanometer. So 420 base pair का अगर DNA है, which is having 420 helical turns, simply multiply 420 with 0.34. Are you getting it? That will give you that what is the length of that particular DNA. Are you getting it? So if you know what are the physico-chemical property of the DNA, you know the conversion of the base pairs to kilo base pairs, etc. You will be able to answer this type of questions. Are you getting it? If somebody will ask you, how many base pair will be present in 1 meter of DNA? Convert 1 meter of DNA into the nanometers. Are you getting it? And find out that if 0.34 nanometer of DNA contains 10 base pairs, then 10 raised to 9 nanometer contains how many base pairs? So 10 raised to 9 divided by 0.34. Are you getting it? So these kind of questions related to the structure of DNA is generally being asked in the various competitive examinations. Now we know that DNA, it is a macromolecule, and you know that it is made up of nucleotides, strands of nucleotides. What do you mean by nucleotide here? Nucleotide means nitrogen base plus deoxyribose plus phosphate. You know nitrogen base, nucleoside and nucleotide. So nucleotide means nitrogen base, deoxyribose sugar and phosphate. Now here, as far as the number of base pair involved in the formation of the number of turns in the DNA, many different questions can be asked. I have given one example over here. For example, the linear sequence of the bases provides actual genetic information. Now we know that there are only 4 bases as far as the structure of the DNA is concerned, which are complementary to each other. For example, adenine, guanine, cytosine and thymine. So if I want to construct the DNA of X chain length, how many different configurations or how many different combinations are possible? This question can be asked. If I have 4 different nitrogen bases, we know that DNA contains adenine, cytosine, thymine and guanine, and from that, if I want to construct the DNA chain of X length, how many different combinations are possible? So here you have to remember this formula, 4 raised to X. That formula you need to remember, where X is the number of configurations in terms of the length which you want to form. यहाँ पे मैंने एक एग्जाम्पल दिया हुआ है. For example, chain of 2 bases long DNA using 4 possible bases, then how many combinations are possible? 4 raised to 2, this is equal to 16 possible configurations of DNA are possible. So these kind of questions can also be asked. These 16 different combinations are given over here, that how you can arrange 4 different nitrogen bases to form the DNA with a chain length of 2 bases, and these are the different configuration in which we can obtain 16 possible different configuration by which we can obtain the DNA with the chain length of 2 bases. Same type of question can be asked from the proteins and amino acids, that if you have 20 amino acids, how many different possible tripeptide can be formed from the 20 amino acids? So very simple, 20 raised to X. Are you getting it? These type of questions are being asked in the competitive examination, that if you have 20 amino acids, how many tripeptide is possible? If you have 20 amino acids, you know that total number of amino acids are 20, from which protein is being synthesized. If you want to synthesize the tripeptide, 20 raised to 4, tetrapeptide 20 raised to 3, and so on and so forth. Similarly, they can give you the question where they will ask you that if I have 4 different amino acids, how many different possible peptides are formed? 4 factorial. The answer will be 4 factorial. That is 4 into 3 into 2 into 1. That is 24. Are you getting it? So these kind of questions are generally asked. Another important topic from which the question is being asked is the Chergov's rule. Are you getting it? We know that there is a complementary structure of the DNA which allow precise duplication. One strand determines the sequence of the other, and there is a hydrogen bond between the adenine and thymine and guanine and cytosine. This fact is known to you. But you tell me that whether the RNA will follow the Chergov's rule or not. Chergov's rule is applicable to all the nucleic acids, both types of nucleic acids, or it is applicable only to DNA? It is applicable only to DNA because DNA is a double helical structure. RNA is not a double helical structure, it is a single stranded nucleic acid. It may form the stem and loop structure when it will undergo complementary base pairing with its own strand, but otherwise this Chergov's rule is being followed by the DNA only. Are you getting it? Now, what is the Chergov's rule? You must be knowing that total amount of is equivalent to the total amount of thymine which is present in the DNA, and total amount of cytosine will be equivalent to the total amount of guanine which are present into the DNA. So, by and large, we can say that total amount of purine will be equivalent to total amount of pyrimidine. So if you take the ratio of total amount of purine to total amount of pyrimidine, that will be 1. So on the basis of this, many questions are being asked. For example, they can ask you that if we have the DNA molecule in which 50% of adenine is present, what will be the percentage of thymine in that? What will be the percentage of guanine in that DNA molecule? For example, I have the DNA molecule. The DNA molecule has 50% of adenine. Now I want to find out how many percentage of guanine will be present in this DNA. So if you know the Chergov's rule, you will be able to answer this. What is the complementary base pair for A? What is the complementary base pair for A? T is the complementary base pair. So it means, as per the Chergov's rule, if 50% is A, then there must be 50% of T. So total 100% हो गया. It means this DNA does not contain guanine and cytosine. 0% of guanine will be there. Are you getting it? Total amount of adenine should be equal to total amount of thymine present in the double helix. Are you getting it? So that's why, see, this, if you don't know the Chergov's rule, then you will end up with the wrong answer. Are you getting it? So you should see that as far as the 4 different nitrogen bases are concerned, what should be the total percentage? It should be 100%. It should be 100%. It can never be more than 100%. Are you getting it? So that's why here, if adenine is 50%, its complementary thymine will be 50%. So 100% तो हो गया. It means this DNA contains only 80 base pairs. Are you getting it? Okay, let us discuss some of the model questions, and I now expect that you will be able to discuss these questions. These all questions are being asked in the previous JNU examination. Are you getting it? Okay. First question is that if DNA molecule contains 30% of thymine, if DNA molecule contains 30% of thymine, what will be the percentage of guanine in that DNA molecule? समझ में आ गया कैसे? ओके, 30% थाइमिन है तो एडेनिन होगा 30%, टोटल 60% हो गया. अब बचा कितना? 40%. दैट इज टू बी इक्वली डिस्ट्रीब्यूटेड बिटवीन द ग्वानिन एंड साइटोसिन. दैट इज टू बी equally distributed between the guanine and cytosine. So 20% will be guanine and 20% will be cytosine. This we have already discussed, I think, that if the DNA molecule has 4200 base pairs, how many helical turn it contains? We know, as we have discussed earlier, that 1 ton of DNA has 10 base pairs. Actually it is 10.4, but for the calculation purpose let us consider that it has 10 base pairs. Are you getting it? So if 10 base pairs produce 1 helical turn, 4200 base pairs produce how many helical turns? So 4200 divided by 10, that comes to 420. And if somebody will ask you that what will be the length of this DNA in terms of the nanometer, multiply this 420 with 0.34, because we know that the pitch size of the DNA is 0.34. The 1 helical turn, uska jo size hai, that is 0.34. So multiply 420 with 0.34, that will give you an idea regarding the length of the DNA. However, this is theoretical length which you are calculating. You know that DNA has the capacity to undergo different kind of packaging. Are you getting it? It may undergo packaging, especially in the eukaryotic cell. You know about the histone proteins and nucleosomes and etc. Are you getting it? So whatever the length which we are calculating here, that is of unpackaged DNA. Are you getting it? When we undergo packaging, the length will reduce. You have to unfold the DNA if you want to find out what is the exact length of the DNA. Do you know how much DNA your one cell contains, which you cannot observe with the naked eye? It contains 2 meters of DNA. 2 meters of DNA, but due to the extensive packaging of the DNA, it is being accommodated so nicely in the cell. Are you getting it? So that's why 2 meters of DNA is being packaged in the cell which you cannot observe with the naked eye. That is because of the packaging. Are you getting it? At present we have the packaging of up to the ratio of 50. We have not been able to resolve what are the further packaging ratio of the DNA. Whatever the data which you have, or whatever the material or whatever the content which you have studied up to the scaffolding, that's up to the scaffolding, up to the formation of the nucleosome, it refers to the packaging of the DNA up to the ratio 50. There are many other packaging configurations which we have not yet find out that how many different packaging are there. Besides the scaffolding, it further folds in order to get accommodate itself within a cell. Okay, how many different possible combinations? How many different possible combinations of DNA helix is possible with 4 different nitrogen bases? How will you solve this problem? We have discussed. आपको DNA का चेन लेंथ कितने का बनाना है, जिसके अंदर 4 different with 4 different nitrogen bases. How many different combinations are possible? 4:4, 4:4, 4:x का फॉर्मूला मैंने आपको दिखाया था. Yes or no? हमने क्या डिस्कस किया था कि if I want to synthesize the DNA which is 2 base pair long, if I want to synthesize the DNA which is 2 base pair long, here I want to synthesize the DNA which contains with 4 base pairs, which contains 4 nucleotides, 4 in configurations, different configurations. The length of the DNA will be 4 base pairs. So 4:x, 4:4. So that comes to 256. That comes to 256. So here 4 base pairs represents the length of the DNA from 4 different nitrogen bases. 4 base pairs represents the length of the DNA which is to be synthesized from the 4 different nitrogen bases. So here you apply the formula 4:x, where x is the chain length of the DNA. where x will be the chain length of the DNA. जितने लेंथ का DNA आपको बनाना है, जितने बेस पेयर के लेंथ का आपको DNA बनाना है, that is represented by x. जैसा आपने माइक्रोबियल काइनेटिक्स पढ़ा होगा कि माइक्रोबियल काइनेटिक्स में आप 2n का फॉर्मूला यूज करते हैं. जैसा नो फॉर एग्जांपल अगर मैं आपको ऐसा पूछूं if I will inoculate one bacteria in a nutrient broth, after 5 generations how many bacteria will be there? 2^5, 32 bacteria will be there. Similarly here also, if because why there is a 2n, why there is a 2n or 2x? Because after successive cell division or the binary fission, bacteria doubles. That's why 2n. Here we know that there are 4 nitrogen bases from which we are going to synthesize possible configurations of DNA, and that's why 4x. Are you getting it? So those kind of questions can be asked from the structure of the DNA. Okay, now that was the very basic concept regarding the DNA as far as the competitive examination is concerned. Now we will be focusing on the different modes of gene transfer. By and large, there are 2 different modes of gene transfer. One is the vertical mode of gene transfer, and second is horizontal mode of gene transfer. Vertical gene transfer means that the DNA is inherited from the parent to the daughter. Are you getting it? This kind of gene transfer is known as vertical gene transfer, where the DNA of the parent is inherited into the daughter cells. It means here the transfer of DNA takes place between the 2 different generations. Yes or no? Are you getting it? When we talk about the vertical gene transfer, the DNA of the parent cell is getting into the daughter cells. So parent cell represents one generation, the daughter cell represents another generation. So in vertical transfer of DNA, the DNA is being transferred from the parents to its progeny, which represents 2 different generations. Are you agree with me? Okay. Now as far as the horizontal gene transfer are concerned, horizontal gene transfer takes place, the genetic information can be transferred between the cells of the same generation. Are you getting it? The transfer of DNA between the cells of the same generations. You know the various methods of horizontal gene transfer. Can you give me one example? Of horizontal gene transfer? Anyone? Conjugation, transformation, transduction, transposition. These all are the examples of the horizontal gene transfer, where the genetic information can be transferred between the cells of the same generation. Now, when this genetic transfer takes place between the cells of the same generation, how we will come to know that this genetic exchange has taken place? Unless until the genetic information which has been transferred, it has some selectable marker and it will get integrated into the DNA of the host in which it has been entered. Are you getting it? So just entry is not sufficient. The functional integration of the DNA into the genome of the host is also as much important, and that is being done by the process of recombination. That is being done by the process of recombination. You must have studied different types of recombination, right? Can you enlist them? Can you enlist different types of recombination? Homologous recombination, that is holiday model, which you have studied. Heterologous recombination and site specific recombination, which you have studied during your repair mechanism. आपने पढ़ा है ना repair mechanism, बराबर? DNA UV radiation से अगर डैमेज हो जाता है तो you have light repair and dark repair. In dark repair you have studied site specific recombination repair. Are you getting it? So this recombination is the important phenomena which will allow integration of the exogenome, that is the gene which is coming from the another organism or the environment, into the endogenome, that is the DNA of the host itself. Are you getting it? Now whether after the recombination, whether the organism which has received this DNA will get converted into the transformants, conjugant or transductant of your required marker or of your required trait, that will depend upon that how this recombinant is resolved. For example, you know the recombination. Recombination also takes place in the eukaryotes. Are you getting it? For example, you have studied different stages of cell cycle, mitosis, meiosis. In meiosis, you must have studied the Pachytene stage. So during the Pachytene stage of the meiosis, the crossing over takes place. So for the recombination, either the DNA is getting entered into the host cell by transformation, conjugation, transduction or transposition. It will not express its traits, it will not express its markers unless until it will get integrated into the endogenome. Endogenome means the genome of the host. The genome of the host in which the foreign DNA has entered. Are you getting it? Okay. Now you know the process of recombination. You know the process of recombination. What is the maximum frequency? What is the maximum frequency of the recombination which is being taking place in 50%? Why the answer is 50%? What is the frequency of crossing over? What is the frequency of crossing over? Because for example, if these are two different DNA strands, two duplexes are there, is it visible? Now it is visible. Okay. Now this is okay. See, this is 5 prime to 3 prime, 3 prime to 5 prime. When the recombination takes place, first of all the synapsis occurs. It means that two duplexes will align together in a row, right? You are aware of this fact, yes or no? So two duplexes, they will align together. The nick will be introduced. The nick will be introduced, and one non-sister chromatids. This is the non-sister chromatids for this duplex, and this is the non-sister chromatid for this duplex. So they will approach together. Which protein is involved here? Which protein is involved to form the crossover point? RAG-A protein. RAG-A protein, because RAG-A protein, it has the DNA binding ability, it can bind to the single strand of the DNA, and it has the ATPase activity. So whatever the energy which will be released during the recombination due to the ATPase activity, that will be used to stretch the DNA, both the non-sister chromatids, in order to form the point known as, this point is known as junction, or you can say crossover holiday junction or crossover point. Okay. Now here the answer comes, as one of your classmates has told, that the maximum frequency of recombination is 50%. See, the crossing over frequency can be 100% maximum, but the recombination frequency can never be more than 50%. Are you getting it? It means after the recombination you will always have 50% of cells which are of recombinant type and 50% of cells which are of parental type. This is the best possible combination which you can get after any recombination event. So it indicates that the maximum recombinant frequency is 50%. It is 50%. Now why it is so? You know that crossover is 100%. Crossover frequency is 100%. If the DNA wants to undergo recombination, either there is the process of transformation, transduction or conjugation, it must form the crossover point. It is the prerequisite. It is the prerequisite for the recombination to take place. Now, the question is that how this crossover point is getting resolved. Whether these two invading strands are being cut or two non-invading strands are being cut. Are you getting it? તમે બે અલગ અલગ દોરીની જો ગાંઠ મારેલી હોય અને એને ગાંઠમાંથી જ કાપી નાખો તો એ બંને દોરી પાછી અલગ થઈ જવાની એના મૂળ સ્વરૂપમાં. યસ ઓર નો? પણ તમે જ્યાં ગાંઠ નથી ત્યાંથી તમે કાપશો તો જ તમને બંને એક દોર માની લો કે મારી પાસે બે અલગ અલગ થ્રેડ છે, એક લાલ કલરનો થ્રેડ છે, એક લીલા કલરનો થ્રેડ છે, બંનેને મેં ગાંઠ મારી. હવે ગાંઠમાંથી જો હું એને કાપી નાખું એ થ્રેડને તો મારી પાસે શું વધવાનું? રીકોમ્બિનન્ટ મળવાનું મને નહીં મળવાનું. મારી પાસે પાછું જેવું હતું એવું જ મળવાનું, એક લાલ દોરી ને એક લીલી દોરી. પણ હું એને નોન ઇન્વેડિંગ જ્યાં ગાંઠ નથી મારી ત્યાંથી હું કટ કરીશ તો મને રીકોમ્બિનન્ટ મળશે. અડધી દોરી લાલ હશે, અડધી દોરી લીલી હશે, ગ્રીન કલરની હશે. તો સિમિલરલી હિયર ઓલ્સો that how this crossover point is being resolved, that will determine that whether you will end up with the recombinants or you will end up with the parental types. If two invading strands are being cut, then there is no recombinant formation. Whatever the progeny will be there, it will be of parental type. क्योंकि जहाँ पे recombination शुरू हुआ था उसी point को आपने काट दिया, so it cannot progress further. For the exchange of DNA, exchange of information between the two non-sister chromatids, there must be the migration of the crossover point. Yes or no? There must be the migration of the crossover point, which is known as branch migration. If there is no branch migration, there is no exchange of information. So if you cut this branch, there is no migration, there is no exchange of information, and that's why you will come up with the parental type of cells. Are you getting it? Okay. But if this crossover point is not being cut, if two non-invading strands are being cut, then this crossover point is intact. It can migrate, it can exchange the information, and that's why you will have the parental type, or you will have the recombinant type of the DNA. So that's why the probability that after the formation of the crossover point, what will happen? Whether the invading strands are being cut or whether the non-invading strands are being cut, that will determine the formation of the recombinant or the parental type. और उसकी possibility कितनी है? 50%. उसकी possibility कितनी है? 50%. 50% chances are there that whether the invading strands are being cut or non-invading strands are being cut. So that's why the recombinant will form only if after the crossing over the non-invading strands are being cut. You know that there are three different proteins which are involved in the resolution of the recombinant: RUV, A, B and C. You must have studied. તમે ભણ્યા છો, holiday model ભણી ગયા છો ને? Holiday model of recombination. You must have studied the different proteins involved in the process of recombination. RUVA, RAC BCD protein, RUV ABC protein. So this RUV ABC protein complex is responsible for the resolution of the recombinant, and that will decide that whether it will cut the invading strands into from the crossing over point or from the region where crossing over has not taken place. If there is a resolution of the recombinant from those areas where crossing over has not taken place, in that case you will have the formation of the recombinant. If it is from the area where the crossing over has taken place, in that case we have no formation of the recombinant. We have the parental type of progeny. So what is the probability of this? The probability of this is 50%. 50% chances are there that RUV ABC protein complex will cut the invading strands, and 50% possibilities are there that this RUV ABC protein complex will cut the non-invading invading strands. So that's why whether the recombinant will be formed or not, the probability is 50%. If I will ask you that between the two DNA molecules, if the frequency of crossover is 30%, What will be the frequency of recombination? What will be the frequency of recombination? બે DNA મોલેક્યુલ વચ્ચેની જો ક્રોસઓવર ફ્રીક્વન્સી 30% છે તો recombination frequency કેટલી હશે? 15% and vice versa. Yes or no? So on the basis of this, then you can use the formula also that the number of recombinants divided by number of recombinants plus parental type into 100. So this will also give you the recombination frequency between the two DNA molecules in terms of the percentage. You can also use this formula. हालांकि जरूरत नहीं है उसकी, ये आपको formula कब use करना है? जब आपको इस तरीके का data दिया गया है that if I will have two different organisms in which between the DNA molecules the recombination has been performed and we have got two recombinants and we have got two parental types, what will be the frequency of recombination? So 2 upon 4 into 100, that is 50%. Are you getting it? Are you getting the point? Are you getting the concept? Okay. So we will one by one discuss all these different processes, whether it is a transformation, translation or conjugation, and this is the very basis of any this process, that the functional integration of the DNA molecule into the endogenome, that is into the host, unless until whatever the traits, whatever the markers which are carried by this DNA fragment will not be expressed, you will not be able to find out, you will not be able to score whether the transformant has formed, whether the recombinant has formed, whether the conjugant has formed, because that has to be propagated within the progeny, and that is only possible if it gets integrated into the chromosomal DNA of the host organism. So we will start with the bacterial transformation. Bacterial transformation, you know the definition, right? You have studied all these things. Bacterial transformation means the genes which are transferred as naked DNA. Genes are transferred as a naked DNA. Here there is no physical contact is required. You must be aware with the Griffith experiment, right? You must have studied the Griffith experiment. I don't need to explain the Griffith experiment. We will focus on the mechanism of the transformation. So the genes which are transferred as a naked DNA, and it is not just merely the transfer. After the transfer, this naked DNA must get integrated into the host DNA in order to propagate further in the subsequent generation, and then only we will have the formation of the transformants. Now generally it can occur between the unrelated genus and species. It is not always true, however. If we talk about the gram-positive bacteria, then it may be true that gram-positive bacteria, for example, Streptococcus pneumoniae, can take up any DNA. It does not have any reservation that I will take up the DNA only from my own species. But if we talk about the gram-negative bacteria, for example, Haemophilus influenza, it will take up the DNA only from its own species. Are you getting it? This is the very basic difference between the gram-positive and gram-negative bacteria. So whatever we are discussing, the experimental evidences related to all these different characteristics may be one of the area from which the question can be asked. Are you getting it? For example, आपको ऐसा एक diagram दे दिया गया in case of the transformation. As far as the specificity of transformation is concerned, what we have discussed here, that the transformation can occur between the unrelated genus or species. This is true for the gram-positive bacteria. It is not true for the gram-negative bacteria. gram negative bacteria can take up only that DNA which is from its own species. अब मान लो आपको एक ग्राफ दिया गया है, आपको एक डायग्राम दिया गया है, उसके ऊपर से आपको फाइंड आउट करना है that whether this uptake is with respect to gram positive bacteria or gram negative bacteria. For example, DNA uptake rate versus concentration of DNA. I am depicting two different graphs. You have to tell me which one is for gram positive, which one is for gram negative. DNA concentration. The DNA which here I am using is calf thymus DNA. And rate of DNA uptake. Rate of DNA uptake. Again, same on the x-axis, same on the y-axis. You tell me which represents the DNA uptake in gram positive bacteria, which represents DNA uptake in gram negative bacteria. Yes, left hand side is, ये left hand side वाला है वो gram positive है because it will accept the DNA regardless of the source. Are you getting it? ये जो statement लिखा है ना, it can occur between the unrelated genus of the or the species. So if you increase the DNA concentration, the uptake rate will also increase. The DNA uptake rate will also increase because the gram positive bacteria will accept the DNA regardless of the source. Even if you use the calf thymus DNA, it will be able to accept. It will be able to accept the calf thymus DNA. But however, whether this DNA will undergo recombination and form the transformant, it is a different issue. Are you getting it? DNA अपटेक हो गया, उसका मतलब ऐसा नहीं है कि वो रीइंटीग्रेट हो जाएगा एंडोजीनोट में और रिकॉम्बिनेंट बनेगा. What we are talking about rate of DNA uptake. As far as the uptake of the DNA is concerned, the gram positive bacteria is not specific. It can accept any DNA regardless of the source. As far as the gram negative bacteria is concerned, there is no increase in the rate of the DNA uptake. तो ऐसा अगर आपको ग्राफ दे दिया गया है और आपको पूछा गया है that which of the following indicates the DNA uptake in which kind of bacteria, तो आपको पता होना चाहिए that in case of the gram negative bacteria, it will accept DNA only from the same species because it is a sequence specific. There are certain specific sequence of 15 different nucleotides. If that sequence is not present on the DNA, it will not be taken inside by the gram negative bacteria. So that's why, okay, now also from the given population of the bacteria, all the cells cannot pick up the foreign DNA. Only the competent cells can pick up the DNA. What do you mean by competency? What do you mean by competence? What do you mean by competence? How can it be acquired? Yes. Okay, better. What do you mean by competence? Competence means it is an ability. Competence means it is an ability of the organism to take up the DNA. Barabar? Now, he was talking about the stage. At what stage this competence is developed? If we talk about the gram positive bacteria and gram negative bacteria, both of them develops competence, but their method of developing competence is different. The gram positive bacteria, they will acquire the competence by virtue of competence factors which are protein in nature and which are extracellular in nature. Which are protein in nature and which are extracellular in nature. Now, since they are protein in nature, they must be the primary metabolites of the organism. I think you are aware with this terminology, primary metabolites, secondary metabolites. Are you getting it? So if you look at the bacterial growth curve, how many stages are there? Time versus number of bacteria. How many stages are there? Lag, log, stationary and decline. Right? This is the typical bacterial growth curve. Now, since these competence factors are protein in nature. They are protein in nature and they are extracellular. So, since they are primary metabolites, since they are protein in nature, they will be produced during the log phase of the bacterial growth. As he mentioned that they are produced at some growth stage. So, it is produced during the log phase of the particular bacterial growth curve. Now, it doesn't mean that when it will produce the competent cell, the bacteria will become competent. It is not like that. Are you getting it? It will produce the competence factor, and when the concentration of the competence factor will reach to certain particular concentration, the bacterial cell will become competent. Because the competence factor, they are the primary metabolites and their concentration is dependent on the population density. It dependent upon the population density. So the cell will become competent only when they will cross some critical concentration of the competence factor. Competence factor બનવાનું ચાલુ થઈ જશે અહીંયા log phase થી જ, પણ સેલ કઈ તરત competence factor બન્યો એટલે competent થઈ જશે એવું નથી. When it will become competent? When it will become competent? At the end of the log phase. Why at the end of the log phase? Why at the end of the log phase? Because at the end of the log phase, we will have some critical concentration of the competence factor. Competence factor are the primary metabolites. They will depend upon the population density of the culture, and that's why at this particular stage, if I will measure the number of transformants